pleasure to introduce. Okay. It's my great pleasure to introduce my friend Lai Guang, and I think Lai Guang's career really illustrated the definition of being international. So as Malaysia, I think Lai Guang actually obtained his PhD degree in Guan Institute of Medical Research in Sydney, Australia, and then he did the postdoc in Singapore, and then he moved on for another postdoc training actually in Wistar Institute in Philly, USA. And then actually he moved to Singapore and joining Singapore Immunology Network in 2009 and has been there ever since. And Lai Guang's work actually focuses on one a really interesting cell type, neutrophils. And I think, in my opinion, neutrophils are certainly understudied, probably one of the most understudied immune cell populations, and giving their importance and sheer number. And I think Lai Guang addressed three critical questions that neutrophils would have asked themselves. So where am I? Where I'm coming from, and what I'm doing. So, I think without further ado, I would give the mic to Lai Guang, and welcome, Lai Guang.
Right. Thank you, Xiaoyu. So, first of all, I definitely would like to thank Xiaoyu for kind of getting me into this fraternity, like a special VIP club. That is what I told my lab, that I'm so happy that I was allowed to join the. To me, it's a VIP club that I can discuss with many of my colleagues online, and unfortunately online now, and hopefully there will be a day that we all can meet together much easier compared to what we have to do normally now, like through Zoom or Team. And for today, the title of my talk is Neutrophil, and I hope that by the end of this talk, I'll be able to convince you that neutrophil are important, and they don't work alone. They work with a group. So that's the reason why I kind of get these ideas of having this title everywhere I go, calling neutrophil the power of many. And it's a little bit of it's kind of inspired by Ido Amit. So Ido Amit is like going everywhere, giving a talk called the power of one, means that the single cell. So I kind of I was in one of the meeting together with him. Then I kind of just want to joke with him, so I use the title "The Power of Many" instead of the power of one, and that is the reason why. And hopefully, more importantly, I would be able to illustrate the features, the special features of neutrophil after this talk. So as you definitely will know that neutrophil is represent the 60 to 70% of our circulating leukocyte in human, and is about 20 to 30% in mice. And they play a major role in defense against microbial pathogens, and they are the first blood-borne cell to be recruited to the inflamed or infected site. And how these cells carry out their function is by releasing the mediators and enzyme that will not only attack the microbial pathogens, it will also damage the surrounding healthy tissue. Because of this reason, their production, release, migrations, and clearance are highly regulated. So I just want to quickly draw your attention to one of these intravital imaging we performed years ago. This is I should say the videos of injury induced by the laser in a neutrophil reporter mouse, a lysozyme GFP mice. And you can see that with the laser injury in the middle here, how the neutrophil is being recruited to this site, to this injury site, and this green cell represent the neutrophil. And two features you can immediately realize. is that they kind of work in a large number. It's not just one or two neutrophil, it's a group of neutrophil going there. And the other major feature is that it's quite rapid, so they are quite rapidly be mobilized. However, this may actually pose a problem for our system. As you may know, that neutrophil are relatively short-lived compared to many other leukocytes. Their half-life is about 6 to 12 hours in circulations in both mouse and human. And to meet such demand, high demand for neutrophil turnover, because it's a high turnover, and to meet this demand, the neutrophil production, in fact, represent the major activities of the bone marrow. In fact, 2/3 of these hematopoiesis processes is devoted to myelopoiesis. So when we started this project a few years back, how we kind of asked question is that if we need so many neutrophil and to better understand neutrophil biology, so it is important for us to understand how they are developed. So when we look back at the literatures at that time, we know that neutrophil is derived from GMP. However, we also notice that there is a big gap in the knowledge, whereby it is unclear what are the precursor or progenitors of neutrophil. So that wasn't clear at that time. It's not that like saying that there's no information about it. There are ways to characterize them based on this very classical hematological um, approach by dividing a neutrophil developmental stage into myeloblast, promyelocyte, myelocyte, metamyelocyte, and band cell. But this kind of characterizations method is purely based on morphology, as well as the nuclear cytoplasm ratios and type of the granules. And this type of method is not ideal in a sense that it's very time consuming, and it may also have reproducibility issues depending on the experience of the operators or the researchers. And more importantly, it doesn't allow downstream analysis, because like once you put it on the slide using the Ginza stain, then that will be the final outcome. There's no way to isolate them for downstream analysis. So for us, then we ask the question that is there a way that perhaps we can identify the precursor? How can we identify the precursor? Is there a specific characteristic that we can use as a marker to identify the precursor? So then we kind of went back to more like a hematopoiesis review papers and tried to understand how all these things happen. And we realized that all the HPC means that the progenitor cell or precursor cell, they are highly proliferative and low cell renewal, as compared to the HSC, which is like they are highly they are high in self renewal capacity and low proliferations. And mature leukocyte, which is known to be low proliferations and low cell renewal, depending on some cell type. There's some exception, but generally they are low in both these aspects. So for us, we thought, okay, if let's say that the progenitor cell precursor, they are highly proliferative, can we use the proliferations as a marker to help us to identify such unknown or putative neutrophil precursor? So so using the proliferation as a marker, so we also want to we are hoping that at that time we are hoping that we will be able then to identify the surface marker. By having this surface marker, we can then isolate the precursor or the progenitors of neutrophil for downstream analysis and try to better understand the ontogeny of neutrophil. So how do we do that? So we made use of the mass cytometry technology or CyTOF technology by staining the bone marrow cell with IDU. So IDU essentially work in a similar fashion with BRDU, which will tag the proliferating cell. So what we have done here is using the cytoph approach and stain with 40 different markers together with the IDU. Just to quickly give a kind of a brief overview of the cytoph technology. So essentially this cytoph technology works similar as a flow cytometry. The advantage of using cytoph is that all the antibody is tagged with a metal. So each metal will have different mass, and by doing this mass cytometry, you will then distinguish each of the antibody based on the mass of the metal, instead of like a conventional flow cytometry whereby they may have be some spectra overlapping, so you cannot distinguish each color so well. So by having this kind of approach, the advantage is that we can look at multiple marker, in this case, 40 of them simultaneously together with the IDU. And as you can see from this approach, we can then uh, separate the populations into IDU negative, which is the non-proliferative cell, and IDU positive, which are the proliferative cell. And for in our case for these studies, we mainly focus on neutrophil. So by using this kind of approach, we can then look at the differentially expressed surface marker between the IDU positive, the putative precursor for neutrophil, and the IDU negative neutrophil, which are the non-proliferative neutrophil, which we will consider them as the mature cell. So you can see that these are the some of the markers that we can identify. So from here, want to uh, we then want to validate it using conventional flow cytometry. So before we jump into doing flow cytometry, we kind of want to have another layers of validation. So what we did here is we made use of the FTSE reporter mouse. So this mouse, the unique feature of this mouse is that when the cell are dividing, they will become green. And when they stop dividing, they will become red, depending on which phase they are in their cell cycle. So using this particular reporter mice in a way that we will be able to tag all the proliferative cell and test validate our markers that are identified from our cytoph experiment. And as you can see from here, when we check this mouse, it is like what has been reported. All the precursor cell, they express the GFP, which means they are in proliferations. All the mature cell, essentially there are none of them expressing the GFP markers as well as the stem cell. So it kind of suggesting that this is a good model to tag the proliferating cell. And using this mouse model, we can then gate it on the FTSE positive and negative cell. And together with our markers, we then validated that the putative neutrophil precursor express CXCR4 and c-KIT, and they are low in CXCR2 expression and high in CXCR4, as you can see here. And they are also they are also Ly6G low. So in that sense, that we so-called identify the putative neutrophil precursor, which are c-KIT positive, CXCR4 high, Ly6G low, and CXCR2 negative. So since this paper was published several years ago, I won't go into much detail. So I just want to quickly summarize using a cytoph approach and multi-dimensional flow cytometry, as well as some of these spatial immune mapping methods, which are reported in our papers. We essentially we identified the three subsets of neutrophil in the bone marrow, which we name it pre-new, which is the precursor of neutrophil. They are proliferative, and the mature neutrophil, which are non-proliferative, and the mature neutrophil that resemble those that is already been released in the blood. So these are the three subsets of bone marrow neutrophil that we have identified and can clearly distinguish them based on several markers. So that was all well, but one of the major things that my student found at that time that also puzzled us, and which also leads to us to continue to study more about this along this line, which was the fact that when we isolated the pre-new and cultured them in vitro, they can only form a limited colonies. They do not form multiple, like kind of big colonies like those you can observe from the GMP cell, which is the granulocyte, monocyte progenitors. So this kind of raised the questions that if pre-new can only form a limited colonies, how can these precursor cell give rise to enough numbers of neutrophil in terms of the immature, then subsequently the mature cell? Because as you can see, the immature and both immature and mature cell, they are not proliferative anymore. So that was our question that kind of puzzled us, and because of that, we hypothesized that perhaps there is another population or more than one population that we still do not know, and they could fall in the current paradigm of how GMP has been described. So perhaps, in other words, that perhaps GMP consists of committed neutrophil progenitors that we still do not know, because that is how GMP, based on several markers, is defined. And perhaps this population consists of unknown committed neutrophil progenitors. So that was our kind of starting hypothesis. So how do we do that? So the first thing Emmanuel did, my then student and now postdoc in the lab, what he did was he went to the web and then get the data, single cell data set from Ido Amit's lab, whereby they look at all the myeloid cell, and what he did was extracted the GMP data and perform a t-SNE analysis on them, and immediately what he found is that GMP can essentially separated or clustered into 10 distinct populations, and all these population cluster to so-called like having some signatures of monocytic cell or basophilic or eosinophilic kind of signatures, and these are all the neutrophil and monocyte signatures like in this population, which I will come back later. So based on this analysis, then we are kind of, okay, perhaps we really should look at the GMP much closer. Maybe the missing puzzle is in this population. So the question will be, how do we isolate this subpopulation for downstream analysis and validation? So that's always the key. The single cell data definitely is good and very informative. However, the limitation is that it can't actually help you to do a lot of molecular analysis. So we still need some sort of surface marker to isolate these progenitors if they are there. So with that in mind, we then move to use this legend screen approach uh, developed by Evans Newer Lab together with Foreign Genome Labs just across the hallway. So we kind of tap into this model or approach that they developed, in which we harvested the bone marrow cell and staying with the 13 backbone markers. So these 13 backbone markers will be neutrophil related backbone markers, and we'll leave the PE channel empty so that we can get the BioLegend kit consists of 255 PE conjugated antibody. So we can screen through these 255 antibodies, trying to find the markers for our putative progenitors if they are there within the GMP populations. So that's our approach. By putting them through this screening by flow cytometry, we will then take this data and using the infinity flow algorithm that they developed, in which in short, because I'm not a bioinformatician, so in short, you can imagine that we can take these 255 data, 255 stains antibodies together with the 13 backbone markers. Through this algorithm, it will then kind of compress everything. You can imagine it kind of compresses everything into one single FCS file, which also means that you will have 268 markers in your FCS file. And with that, we can then do analysis. So this is like based on the infinity flow data. We can then gauge on GMP populations. The current definitions of GMP is based on the CD1632 and CD34 expressions. So by gating on the GMP populations, again, we just quickly perform a t-SNE analysis. And this is based on, previously I showed as data based on the transcriptomic data. Now it's based on the protein analysis data, surface protein. You can see that again, the GMP populations can be clustered into 10 distinct populations. So that was kind of very exciting. But however, for us, we want to kind of keep our focus in neutrophil. So we want to find a way, how do we then analyze, or how do we then fish out the surface marker for the progenitors, the putative neutrophil progenitors. So with that, we then move to use the UMAP analysis instead of t-SNE analysis. As you may know that the UMAP analysis, it gives certain advantage over the t-SNE clustering approach. As to UMAP will be able to give you more global view, as well as the interrelationship between each cell populations. So in this case, if you see from our plot here, by performing the UMAP analysis on the infinity flow data set that we have, you can see how this GMP population is connected to the neutrophil, and then the monocyte, and as well as other myeloid cell like DC and basophil. So, but for us, our interest is for the neutrophil. So you can see, interestingly, what kind of make us excited at that time is that we can see this bridge. So we would then kind of focus this region, the so-called bridge region, that to connecting the GMP to the prenew. So we want to see what are the markers that is kind of expressing in these regions, and then perhaps this will then represent the unknown progenitors that is kind of within the GMP population. So just to quickly to the take-home message essentially is that by doing this approach, we can then identify several markers that can help us to fish out these progenitors, and these include CD81, 49A, 106, and CD63. So I won't go into details, but just essentially, I want to quickly point out that having those markers, we can then again, like we can then get on the GMP populations and use and also the Ly6C positive GMP, because a GMP can be separated into Ly6C positive population or the Ly6C negative population, and when you get on these positive populations, we will then be able to see the CMOP, which is the common monocyte progenitors, and based on the markers that we identify, we can also identify the pronew. We call it pronew, neutrophil progenitor. So pronew, and this pronew can be further separated into pronew 1 and 2 based on the 106 and 11B expressions. So essentially, that we think that the pronew 1 is equivalent level to the CMOP in terms of the hierarchy order in the hematopoiesis. So by having all these markers, we can then isolate them, and we perform the bulk RNA-seq analysis. And quite nicely, you can see from this PC analysis, from the Ly6C populations, it is kind of, once you get to the Ly6C positive population, it kind of split into some that will go to the monocyte pathway. Some of them will go to the neutrophil pathway, which we are focusing on. And you can see that the pronew 1 is kind of comes before the pronew 2 and then the prenew. So kind of nicely the sequential order. And more importantly, when we look at the transcription factor, the key transcription factor between the monocyte and neutrophil, you can see that for the monocyte transcription factor, such as the IRF5, 8, and AF4, is upregulated in the CMOB, and it's quite kind of a intermediate in the Ly6C negative GMP, as well as the ProNeu1 is still kind of intermediate. But as soon as the cell are further along the neutrophil lineage or developmental pathway, you can see that it kind of drop down regulate. So it's kind of like when a cell decides to become neutrophil, it kind of down regulate all the monocyte transcription factors. And conversely, for all the neutrophil related transcription factors, it's now upregulated. As soon as the neutrophil decided to become neutrophil, it will then upregulate GF1, CBP epsilon, and per3. And this will kind of downregulate in these monocyte progenitors. So it kind of show that the way that we identify these cells are quite good. And this is just to give you a very kind of good visual view that from all these population, for instance, like CMOB, Ly6C, negative GMP, ProNeu1, ProNeu2, just kind of cluster them based on either the neutrophil related genes or monocyte related genes. Immediately you can see them. It's kind of clustered pretty well. Like for instance, Ly6C GMP is more like it's undecided yet, so it express both the transcription or the factors or the genes that related to either neutrophil or monocyte and others. Whereas once you go towards the neutrophil pathway, all these genes will be upregulated, especially all these neutrophil related gene will be upregulated. And conversely, in the monocyte, like the Ly6C, high GMP, and CMOB, all the monocyte related genes will be enriched. So this kind of give us a confidence that what we isolate is correct, and it's more like a committed progenitors. And one of the major transcription factors that has been reported previously, CEBP epsilon, which has been shown that if those mice that do not have CEBP epsilon, they do not have neutrophil. So that is known from previous studies. But in our case, that since we can delineate the subset pretty well now, we ask the questions that where will this developmental stop happen, and we want to test it out. So in this case, we made use of the CEBP epsilon knockout mouse, and immediately when we look at them, one of the major things is we observe, like all the previous study, definitely there's no mature neutrophil as you compare to the wild type. But because of the way that we can delineate different subsets in bone marrow, we can kind of trace it back all the way that we now know that without the CEBP epsilon, the developmental block kind of stop at PNEU1, and then it cannot go further than PNEU1. So you don't have PNEU2 and onwards. And interestingly, we also observed that this atypical granulocyte, the formation of such atypical granulocyte, because we are using a full knockout now, so we were kind of thinking that perhaps this could be some sort of eosinophil cell that is developing, because we in this mouse full knockout, we also do not observe eosinophil as well. But then recently we generated a flux on CEBP epsilon, and when we cross with the MRP8 mice, what we observe is that this population is still there, and then we were able to get the eosinophil as well, still getting the eosinophil in that mouse. So it's kind of suggesting that CEBP epsilon perhaps may even express as early as in the GMP stage, and will regulate the developments of multiple like the granulocyte, for instance, the eosinophil as well. But the impact is definitely the most severe in the neutrophil. So far I've been kind of pushing the idea of the developmental trajectory of neutrophils from ProNeu1 to 2, and then pre-neu, immature, and mature neutrophil. But we don't have a definitive proof, although based on our transcription studies as well as the knockout study, it seems like that. But to definitively answer the question, especially from the reviewer at the time, we perform this particular study in which we uh, isolated GFP positive proneu 1 and then 45.2 proneu 2 and RFP preneu 1. So essentially, you can consider that we identify neutrophil at their different developmental stage in different color, so that we can kind of combine all of them and transfer them into a recipient and check them simultaneously in the same host over time. So that is what we have done here. And you can see that from these studies, the proneu1 cell that we transfer after 3 days, most of them still kind of develop into immature cell, immature neutrophil, whereas the proneu2, some of them is already gone and developed into mature neutrophil. Whereas the preneu, which is the so-called the most mature one in here, in these three populations, almost like 70% of them is already become mature neutrophil. So it kind of showing that what we think the developmental order is correct, and more importantly, all these three subsets that we transfer, they do not develop into either monocyte or eosinophil in that sense. So they are committed precursor or progenitors of neutrophil once they get into the proneu phase. So this is just kind of a summary of this plot here, just to quickly go through. So we believe that the maturation trajectory is from proneu 1 to 2 and then to preneu. So when you transfer proneu 1, most of them become just immature cell because they're the earliest. So they haven't got enough time to develop further. Whereas when you transfer proneu 2, then you get combinations, and then preneu, most of them already have enough time to become mature neutrophil. So by having all those informations at that time, in fact, I should say that after the first paper in 2018, in which we identified the preneu, and we written a perspective together with Renato and Andreas Hidalgo in Nature Review Immunology, that we written this perspective to propose that a working model of how neutrophil develop. And at that time, we kind of proposed that there is a proneu populations. And with our second paper, the follow-up papers, we then show that indeed this is the case. In the GMP population, there is a proneu 1 population will then develop into proneu 2 and continue on the trajectory. And having said that, while we kind of propose this working model, and I also want to acknowledge that over the past few years, perhaps it's like what Xiaoyu was saying, that neutrophil are one of the most understudied leukocyte in the field, and we are quite kind of happy that for the past few years, there are many labs that is also focusing on studying neutrophil development. For instance, Hendrik's lab in US, Lee Guan's lab in US, and Lo Hongbo Lao's lab in Harvard, as well as Professor Kang's lab in Korea, that are putting effort into trying to understand neutrophil development. And while we may actually, as you can see, that I make a summary of all these studies, as we may all kind of try to name the neutrophil quite differently, and depending on how high resolution you look at them, we kind of name it differently and then characterize them quite differently in some cases. So we are hoping that one day all of us can really sit down and try to unify how we should identify neutrophil subset. And I think this is very, very important work that we have to push forward in the future. By doing so, it will then allow people that are not in our field, not in the neutrophil research field, will be easier to understand our data. Also, that they will be easier for them to perform the experiment and compare to the existing data. Because at this stage, we kind of name neutrophil differently and based on different kind of markers. But slowly, I'm hoping that this will be unified, and then this will then be beneficial for the entire field. So having kind of understand the neutrophil subset in the bone marrow, then we have the question that how would this cell mobilize? So in 2013, years ago, before we started this ontogeny work, we already know that from our intravital imaging studies, we already know that for instance, if you inject GCSF, you will be able to mobilize neutrophil from the bone marrow, as you can see here, into the circulation. These are the sinusoid in the bone marrow, and being all these neutrophil will then be released into the circulation. So in this case, we are using the skull bone approach developed by Uli von Adrian's lab, and you can see that how we can visualize nicely the bone marrow microenvironments. So based on this data, you can definitely see that neutrophil can go out when there's a trigger, and that kind of lead us to try to understand, okay, if that's the case, what kind of population can go out to the circulations. So based on our characterizing, or I should say that the delineations methods, what we found is that if we inject the GCSF, what we found is that normally in the PBS control mouse, you only can see a mature neutrophil, although you still see some sort of like some pre-neutrophil or immature neutrophil, but it's very small frequency. So it's just like stem cell. Like stem cell can be detected in the blood as well, but it's very low frequency. But once you inject GCSF, you will see these very distinct populations of immature cell that are being released into the circulations. And this is just the plot showing that both the immature neutrophil and mature neutrophil. So since that we can see that the immature neutrophil can be released from the bone marrow into the circulations, so then we ask the questions, can this cell be recruited to the inflamed site? Because we all know that neutrophil can be recruited to the inflamed site, but it's still unknown that whether the immature neutrophil are capable of doing so, because they do not express CXCR2, if you remember from my previous slide. So how to address these questions? Then we kind of went back to a very old model that I established during my postdoc days, in which that we can intradermally transfer neutrophil into the mouse ear skin, and then we can induce laser injury, and this will then kind of trigger such a neutrophil recruitment to the injury site in a three-phase cascade. So first is like you can see the scouting. Maybe I should we show this one. So first is like scouting. So few neutrophil will go to the injury site, and with this scouting phase, and then will come the amplification where the neutrophil will call all the other neutrophil into this site, and they form cluster around it, in which we call stabilizing state. So we want to use this model to address whether the immature cell can be recruited to the tissue as well. So how did we do that? So essentially we do a similar approach by just isolating either immature cell or mature neutrophil in two different colors. And in this case, we also isolated a pre-new. So pre-new, we did not detect, like even after GCSF injection, we did not detect more of them in the blood. So we kind of suspect that they may not be so good in their migratory function. So we just want to test it out. So in this case, the green cell either represent the immature cell or the precursor cell, whereas the red cell will represent the mature neutrophil. And if you look at the left panel here, so when you compare the immature and mature neutrophil, mature is in red and then green is immature. both of these cells were able to respond to the injury site and be recruited to this injury site. And whereas for the pre-new, somehow they don't, and they just stay at the depot that we injected them into the skin, and the rest of these mature cell will be recruited to the site. So with that in mind, then we kind of take it like wanted to take it one step further. So we want to ask the question that how is this subset will pan out in a disease model. So in this case, we made use of the KPC autotopic model, which is the pancreatic tumor models, in which we transplanted the tumor cell line into the pancreas in the black 6 mice. And after that, we wait for 6 weeks, 3 to 6 weeks, and we can then look at the blood as well as the tumor. So by doing so, we will then be able to look at the different subset in the blood as well as the tissue. So again, it's very similar to the GCSF data. In the presence of tumor or inflammation, what we found is that what we can see is that there's the presence of immature neutrophil in the blood as compared to the naive mouse. And this immature neutrophil number will increase, especially in those high tumor burden mouse, and as well as and this number also increase in the tissue when we take out the tissue, the tumor from the mouse and do the flow analysis. You can see that those mice that have the high tumor burden, they have more immature neutrophil in the tissue as well, as compared to those that with the low tumor burden. And if we look at it carefully by looking at the tumor burdens, we can really kind of distinguish, like in the blood they will have more immature neutrophil, and when we look at it in a correlative manner, you can see that this is correlating together with the pancreatic tumor weight. So it kind of suggests that perhaps this can be used as a biomarker to detect, in this case, the tumor burden. So just to quickly summarize this part of my talk, is that I hope that I convince you guys that we propose a working model in which GMP can go into pro-new 1, 2, and then go to pre-new, immature, and mature. And more importantly, what I've just shown for the past few slides is that we show that in the resting state, you don't have an immature neutrophil in the blood. But once in the inflammations, you detect this immature neutrophil in the blood as well as in the inflamed tissue of tumor. So this is something that that's kind of very interesting. It also raised the question that whether these immature cell can further develop either in the blood into the mature neutrophil, or they have to go to the tissue and then develop into the mature neutrophil. So this is something that we are trying to address currently. So you may ask me, how would this relevant to in human? So have we done any work in human? So we did, in fact, and how we do that, again, we are using the same approach, which I will not go into details. So we are using the infinity flow as well as the legend screen approach. We took quite a bit of human cord blood, and because it's quite difficult to get bone marrow in Singapore, so we took a few bags of human cord blood, and then we screened through different markers and trying to identify the markers that we can use to distinguish different subsets of neutrophil in human. And in this case, that I just want to summarize that we indeed can use this approach to distinguish on pro-new 1, 2, as well as pre-new and immature and mature neutrophil in human. And using this, we also collaborate with one of our colleagues in SIGN, in which when COVID happened, the pandemic happened 1 year ago, we collected some patient sample. This patient essentially can be stratified into those that are with pneumonia, hypoxia, and those that are with pneumonia, hypoxia, as well as more severe case that they are in the ICU. And the take-home message here is that what we can see from using our methods, we can distinguish that for those that with severe hypoxia and ICU warded patients, they have more immature neutrophil in terms of the total numbers as well as the frequency. So it's kind of suggesting that perhaps, immature neutrophil can be used as a biomarker for a wide range of inflammatory diseases as well as COVID-19 in this case. So just to conclude this part of my talk, with all the published data, I hope that I convince you that neutrophil delineations methods that we devise can provide a framework to better characterize neutrophil in health and disease. And we also identify some of these surface markers for neutrophil subset, and perhaps this can be used to further develop biomarkers in different diseases. And we also show that inflammation induces premature release of immature neutrophil into the circulations. And for the next 5-10 minutes, I want to share some of the unpublished data in my lab, and that's the reason why when Uchan asked me whether this can be recorded, I was kind of thinking about it, but in the end I said okay, it can be recorded. So I hope everyone do not post this to Twitter or something, but if they log in and see my talk, it's totally fine. So the major question is that we're trying to address for this part is about tumor neutrophil. As you may know, that the tumor neutrophil is rather complex, and different people, different lab still kind of name them differently. And predominantly how they are named is based on their functions. For instance, N1, N2, MDSC, or tumor-associated neutrophil. So how they are named is based on, as I mentioned, function. In this case, it's the immunosuppressive functions of this neutrophil in the tumor. So we thought that perhaps with our kind of methods of characterizing neutrophil subset, can we kind of employ it and try to help the field or help ourselves to better understand how these neutrophils should be named or called or characterized even. Because the current way of identifying them or naming them or even characterize them is kind of very unclear, because there's no way to distinguish whether this is a tumor-induced stage or is actually a preferentially recruitment for instance, because of inflammations, as we have shown, you can recruit an immature cell. So could it be a preferential recruitment in the tumor cases? Some of the specific subset of neutrophil being recruited, and this kind of been misidentified as a so-called N1, N2, MDSC, or tumor-associated neutrophil. So for us, we want to address these questions. So how do we do that? We make use of the same sample that I mentioned before, which is the pancreatic tumor sample, the model, and we isolated different subsets of neutrophil based on our methods. In this case, pre-new, immature neutrophil, mature neutrophil, in either spleen, blood, bone marrow of a control mouse or tumor-bearing mice, and as well as those neutrophil in the tumor tissue itself. And after that, we then perform bulk RNA-seq analysis. There's a lot of information in this particular data, and the thing that I really want to point out is that different from what we expect, which is like we expected, like in a tumor-bearing mouse, the neutrophil, it doesn't matter which subset they should be impacted, especially on their transcript level. But when we look at the data, especially in this particular model, we did not see that. In fact, the neutrophil is still clustered according to their maturation status, instead of whether they are actually from bone marrow, blood, or I shouldn't say blood, bone marrow, spleen, in control versus the tumor-bearing mouse. So essentially, the take-home message is that the tumor did not induce so much changes in their signatures, so that all these cells still somehow cluster together based on their maturation status, and you can see from here. And but one of the things that is very obvious is that the tumor neutrophil, they're very distinct from the rest of this neutrophil. So it's kind of suggesting that perhaps there's some reprogramming occurring for this neutrophil within the tumor microenvironment. So with that, we then perform our ataxic, so the bulk RNA ataxic. So with that, what we found is that only tumor neutrophil have the unique open chromatin regions, and it's kind of validating our hypothesis in which that perhaps the reprogramming will occur predominantly in the tumor microenvironment. So to better understand the heterogeneities, neutrophil heterogeneities in tumors, we then perform a single RNA analysis, single RNA seq analysis. And what we did here is that we isolated the CD11B lysis G positive cell. So essentially, it's all neutrophil, positive neutrophils, and we barcoded them so that we can analyze them at one go. Means that we can pull all these cells that are isolated from different organs, including the tumors from different mouse. We can all pull them all together since we barcoded them, and then perform the 10x analysis. And you can see from these data sets that we can get back this kind of a nice trajectory from pre-new all the way to the mature. And again, the tumor neutrophil are quite distinct, faster themselves. And when we perform the RNA velocity analysis, you can also see that it's definitely the trajectory is kind of correct in the sense that it starts from the pre-new stage all the way to the maturation stage. And interestingly, for the tumor neutrophil, it kind of there's two entry points. So it's kind of from here and here.
好，那自动跳到按键叫什么是？
So and this is kind of interesting, and what we propose here that perhaps this represent a mature neutrophil coming in or immature neutrophil coming in and then converge at this point here. So this is something that we're currently investigating. And just to kind of further confirm our data, since we did not perform the single cell RNA-seq analysis ourselves, but we made use of the published data from Lo Hongpo Lao's lab in Nature Immunology. This data set is from Nature Immunology, in which they look at they perform single cell analysis in the bone marrow neutrophil, and this is their data. And if we superimpose their data to ours, you can see that it's superimposed very well in terms of this bone marrow as well as the spleen one. And one of the major things that you can notice is that for the spleen, you can see more of this precursor as well as the immature cell appearing in our sample. So this is kind of indicative of perhaps there is an increased extramedullary hematopoiesis occurring in response to the tumor genesis in these mice. And in the blood, you can see that it's pretty similar as well. And more importantly, we show that this tumor neutrophil again is a very distinct subset that is not found in any of these control analysis. So again, then we continue to work on it. So there's a lot of things that I show here is for discussions, and with those transcriptomic data, as I always mention, as I've been mentioning in my talk, is that for us, we always want to have a way to really study them using surface markers, and then can isolate them for further functional analysis. So we also want to use our legend string infinity flow workflow to identify the markers that are only expressed in the tumor neutrophil, but not on other infiltrating leukocyte. And more importantly, these markers should be induced by the tumor microenvironment, but not due to the inflammatory response. So it's a very unique subset. We are hoping that we can identify such markers. So what we did here is that we isolated the tumor, single cell suspension from this tumor tissue, and we performed the legend screens. And you can see that from these data sets, we can easily split them into the neutrophil, macrophages, and monocyte subset, and these are the dendritic cell subsets. And we can look at 200 over markers. And based on our preliminary analysis, it is kind of very interesting that for the neutrophil subset, this entire subset is a neutrophil subset, but it can easily be subdivided into populations when we look at the immunosuppressive markers as well as the immunoinhibitory markers. So you can see that the CD14, PD-L1, VISTA, 39, it can be easily separated into two populations, as well as some of these immunoinhibitory markers. We are continuing to analyze this data set. And again, like our approach is trying to identify those one or two markers or a group of markers that will only express in the tumor neutrophil, but not in the inflamed host. So that's something that we are currently working on. And finally, I would like to end my talk with just to proposing a working models. Perhaps the neutrophil reprogramming in response to tumor, we would like to think that is mainly happening in the tumor based on our current data and based on our current model. And this reprogramming will kind of promote this neutrophil into a very distinct subsets of neutrophil that can be identified through their transcript profile as well as their surface markers, hopefully. Okay, and I think I think I talked about all this already, but the future plan is to continue to push forward what we've been doing, and I'm hoping that we will contribute more in terms of biomarker development in the context of neutrophils, and we are hoping. My dream is to be able to develop some sort of like a neutrophil-driven therapeutic targets to treat inflammatory related diseases as well as cancer. And obviously, I did not do all this work. I have this group of fantastic people that I love dearly, although I'm quite tough with them, but I love them dearly. And I just want to point out that Max was a PhD student in my lab. Then he started all these neutrophil ontogeny works, which helped to build a very strong platform for my lab to continue to work on. And this work is continued on by Emmanuel, who is now a postdoc in the lab, and Melissa and Leonard is doing the tumor works, and with the help from Changming, Yingrou, as well as Xia Yu, who is now in Tsinghua doing a PhD. She was in my lab for the attachment for a year. She also worked quite closely with some of my postdoc and students, contributed substantially to the project. And also I want to acknowledge Su Zhen, which is a very senior fellow in my lab, who has been helping to train all my people in a very good way. And all the collaborators, my colleagues in SIGN, especially Florent Gino, who is quite instrumental in a lot of things that I do in my career. So thank you. And finally, all the international collaborators that has been supporting me over the years for lots of things that I do. And finally, thank you. I will take any question. Thank you so much.
Hi, yes, thank you, Guan, for such a wonderful talk and for really the comprehensive work during the past 10 plus years, right? for enriching our understanding about fundamental questions of neutrophil biology. I think, after hopefully after his talk, so everyone, is not only more familiar with neutrophils, but also motivated actually to collaborate with Guan, right? To do more studies with neutrophils. And I think there are questions coming in. I already saw questions in the chat box. And but I will start, I'm always confused with the concept of MDSC. So I'm particularly inspired by your the last part of the talk, that you think, yeah, basically all these definitions are really, really confusing. So some people think MDSC, right, they have somehow a distinct lineage. And so I just want to get your expert opinion on what you think about MDSC. So you apparently you prefer to see that neutrophils are actually reprogrammed on site, right, in tumor microenvironment. Yeah.
Yes. Okay. So if based on our current data and also lots of previous work, fantastic previous work that various lab has been publishing, the MDSC, like you said, is very, very confusing. In fact, there are many, recently there are many review paper talking about it again. But now people kind of settle in terms of MDSC can be separated into two major populations, which is the monocytic one, monocytic MDSC, MMDSC, and GMDSC, the granulocyte MDSC. So these are the kind of something that people can agree upon at this stage. And however, the whether they are separate lineage, I think that is a very tough question to address, because there are many people are using different models, so we can only look at one model. So at least in our model, we do not think it's a different lineage. It is from neutrophil that been reprogrammed. Once they get to the neutrophil, they get to the tumor tissue, they have been reprogrammed. So that is our current data is supporting that. And in terms of why there's such confusions, I do think that it comes back to the point whereby there is no good way in the past to distinguish different subsets of neutrophil. And because when you have information, especially in the tumor, in the scenario of when there's a tumor, there's multiple way of recruiting different subsets. Perhaps some precursor or progenitors somehow being released into the blood, and then this has been recruited into the tumor tissue. And when people take it out, they analyze them. In fact, they are just looking at a more immature form or certain subsets of neutrophil. So that's why they call this MDSC differently. Some people call it like 6G low a little bit, CD11B low a little bit, then is the MDSC. But this is too, how can I say, it's not high resolution enough for you to pinpoint. So now what we're trying to do is provide a better solution. We are not saying that we are 100% correct, but we are just proposing that, perhaps we look at it this way, it will help everyone to better understand. So that is something that we're currently doing.
Yeah, I think, certainly, it just makes more sense for me, right? Your model that the neutrophils come in and they are reprogrammed anyway. So I think, I really appreciate, want to show the unpublished data. And also, I want to urge everyone again, right, now to distribute One's unpublished data, right? And
Don't distribute it, but otherwise my postdoc will hate me and my student will hate me. Yeah. So there's a question from the chat box.
Yeah, there is a question from the chat box from Bo, and I think that is, do you think the Sigla-F positive neutrophils are immature neutrophils?
So they are not the immature neutrophil. I do think that they are the reprogrammed one. But in our case, in our model, we did not observe the expression of Sigla-F. Because I do know that this paper, I think when they look at the tumor model, then they have this Sigla-F positive neutrophil. So I do believe that why I kind of call it a tumor reprogrammed neutrophil is in a way that in the future when everyone look at the different models, it kind of perhaps we can call it a secret F tumor reprogrammed neutrophil, or in our case, CD14 reprogrammed tumor reprogrammed neutrophil, and then also provide which tumor you're looking at. And slowly by doing that, I do think that we'll have more clarity on that. So I do not know whether that address all questions. Yeah.
I think we have another question from the chat box from Qiang. If I understand it correctly, it means the neutrophil phenotypes between tumor microenvironment and inflammation. Is that right, Qiang? Or if you want to elaborate your questions a little bit more.
I think I will try to answer it. I think it's what is the difference between the like you said TME and inflammations. I think this is a very good question. So in the past, all these are suppressor neutrophil and things like that. Again, it come back to the question that can people truly distinguish between the neutrophil that somehow reprogrammed by the inflammations or by the tumor itself. So I think there is no proper way of doing that in the past. So these are unpublished data that we are trying to do. In fact, we are trying to address part of it, but not fully on it. But what I can share is that obviously we kind of expected like when we are ready to submit this manuscript in, I don't know when, it will take a long time before we are ready. The reviewer will ask these questions. And what we have done is that we try to look at all these markers that we identify from the legend screen in the tumor tissue, and we try to looking at it like we injected GCSF, we sepsis mouse or infected mice. We have done some of these preliminary work. We do find there's a certain set of these markers only specifically upregulated in a tumor, but not when there is a general inflammation. So I think we are going in the right directions, whereby there's a very distinctive this inflammation versus the tumor. And the other thing is that I also want to mention, we also look at the Luo Hongbo Lao Shi papers data set in which they also look at I do believe E. coli challenge single cell analysis of all the neutrophil. When we compare the E. coli challenge with our tumor neutrophil, again, they're very distinct. So in a way that I think there's a, and also if you look at the supplementary data carefully in the Luo Lao Shi's paper, they also mentioned that the trajectory did not change. There's no appearance of a specific subsets of neutrophil under inflammation. It's just that maybe some population become more or less, but there's not like something like tumor. There's appearance of a totally new cluster. So at least I think that is slowly going to the direction that hopefully we will be able to show this major and very important question that you asked, TME and inflammation.
Yeah. Right. I think this certainly, to me, it looks like in the certainly right direction. I think we have a Chunliang raised hand. Yeah, please go ahead.
Hello. Hi.
Hi, Legran. Hi. Yeah, this is Chunliang. I just finished my training with Paul Fernet. I'm starting my lab in Hong Kong. So in a very nice talk. Yeah.
So you say you're from Paul's lab?
Yes.
So we have to. Yeah, it's a very sad news, as you know.
Yeah. It's very sad. But
so I'm deeply affected by that actually, because I like him a lot, and I met Paul in one of the meetings that we organized in Tianjin when he came to Tianjin. And for some of you guys may not know, Paul passed away 2 or 3 days ago. I think.
Oh really? Well,
Paul Fernet passed away. Yeah.
Oh, okay.
Yeah.
So,
it's kind of a tumor. So it's my question about the also on tumor. So I just read a paper coming not long ago by the lab Becker. They said the human neutrophil, they can kill the human cancer cells. So what's the role of neutrophil? Do they pro-tumor or can they inhibit tumor in mice and also in humans?
Okay. So I think the tumor, at least we don't have exact data of this, so I have to admit, but based on the tracking of, we have done some tracking as well, in terms of looking at the tumor number over time in the, I mean, I should say neutrophil number in tumor over time. And what we found is that there is a kind of a switch, like the number increase and then a changing of different subset. So what we may believe is that during the initial phase, before the neutrophil is being reprogrammed, perhaps they do have some sort of killing effect. But as this tumor persists over time, and then the neutrophil staying in that environment, they will slowly be kind of reprogrammed into a non-killing and perhaps even pro-tumoral kind of neutrophil. So this is as far as we can tell from our data. But you raised a very important question that what we should do is that we should go back to analyze, perform the exact same thing, but in a very early phase of this tumor, not like 6 weeks, because 6 weeks is considered very late already in our tumor. So perhaps we should go back to the very early phase and compare this early and then the late, how they should look. Do they look alike, or based on their transcript, are they even the same cell? This is something that we haven't done, but for multiple reasons. First, I don't have so much money to run the single cell analysis and ataxic, so we have to wait a little bit. Yeah.
Okay, thank you very much.
Yeah, we have another question from Guan. How long can neutrophils survive in tumor microenvironment? Do you think there's enough time for neutrophils to be transformed, affected by tumor? I think it's an interesting question.
Yes. This I hope the Paul will not be my reviewer. So this is ask all the tough question. Good one, but tough. So we are trying to address this already. So I can share like some of the things that we have done as well. Is that we want to ask the question whether they can. First question is like whether they survive longer. So it's a very simple question. So what we did is that we isolated this neutrophil and then just put them into the normal media compared to some neutrophil that we isolated from spleen of even the tumor-bearing mouse, and just to track how long they can live. So indeed, those neutrophils that's already been reprogrammed in this microenvironment, they live longer. So that is one thing that we know. And then second things that we have done is that we co-culture this tumor together with a neutrophil that we isolated from a wild type naive neutrophil. And what we can see is that this cell will upregulate those markers that I was showing in the legend screen, like the CD14, PD-L1, all these markers. They will slowly upregulate these markers. So, and these are the two things that we have done so far, but we are continue to do more studies, and as well as we are hoping that perhaps we want to do some in vitro, and then we also want to do some real-time PCR to kind of type everything up properly. But based on all these, I would say that they would have enough time because they will live longer than normal neutrophil after they are interacting with the tumor.
So maybe a related question is, Lai Guan, do you think these tumor infiltrating neutrophils become resident neutrophils, or they still come and go?
Sure. This is a super fantastic questions that we have been trying very hard to address. I mean, we started off like addressing very similar sort of kind of idea, we want to address like how they're recruited. So we try different sort of isolations and then IV into the mouse and track, but then it's very difficult. We couldn't get it to work. And then we also try to use some reporter mouse, for example, the which we are currently doing, and that may be able to address Xiao Lei, your question is that whether they become resident or they come and go. So what we are using is the mouse that Florent Gidou make, MS4A3 mouse, whereby you can pulse it. So all the GMP derived cell, they will tag with, should be, in this case, RFP. The RFP cell will be, they will express RFP. So you pulse once and then you wait, and so you get one wave of neutrophil that is labeled, and in this tumor mouse, and then we can track that, you know, whether they stay there or they can go into, they are still in the blood or things like that. So these are something that we are doing, but it's a very tricky question to answer. However, this could be helped by what we currently doing, and we already get first sets of data back, which is the spatial transcriptomic. So we took this tumor tissue out, we did the spatial transcriptomic on tumor tissue, and what we can see is that if you remember from our single cell analysis, we could see generally three populations of these tumor neutrophil, three population of them. And in our using this single cell analysis, we kind of map it back to our spatial transcriptomic. Again, we can see these three neutrophil subset is kind of localized very distinctively in the tissue based on their transcript, and all this area that they are residing is very different in terms of their metabolic status. I mean, genes or proliferative genes is very, very different. So this is something that perhaps, hopefully, can help us to address this question.
Yeah, I think this is super interesting, like for the spatial clustering of like different types of neutrophils.
Yes.
Maybe a related question, like you mentioned these tumor neutrophils, when when isolated in vitro, so they have longer lifespan as compared to the circuit. So how much longer? Is that like 1 or 2 fold, or it's a magnitude longer?
So the normal neutrophil will die, like if you culture, it will be like, you know, 1 day plus 2 day would die.
Yes. Yeah.
And then this one can go up to 4 or 5 days.
Wow, that's a pretty significant. Okay, cool. So I think like Dr. Zhou Gang has a question, but I have some other questions I can ask like after maybe Dr. Zhou.
Okay. For Dr. Zhou, questions are, what are the factors? So this is obviously we want to identify that, because if we identify that, that would be, wow, super crazy good. And what we are currently trying to do, especially with the spatial transcriptomic data, we want to, and together with the single cell data that we have, because the spatial transcriptomic data, it consists of everything, inclusive of the tumor transcript, and also other leukocyte transcript in that. But then is a very tough technique to do, so we are still trying to figure our way out. But hopefully, what we can do is, based on this data set, we can perform some sort of a niche net analysis. Means that the ligand receptor analysis, like kind of to predict whether different subsets of these neutrophil, since they are localized differently, whether there is a factors that is kind of specifically important or predicted to be a interacting partner. So this is something that we are aiming to do, but we haven't done it yet. So like I mentioned, this is all very new, unpublished, but I want to show it and then want to get feedback from everyone. So I want to know how people think about this sort of kind of project. So all these questions are very good for me. So I hope I answer your question, Dr. Zhou.
think with no more
Well, I have some questions. I think also Wu Chang probably also have some questions, uh, as usual. So maybe I'm also actually very interested in the first part about the lineage characterization. For the GMP, so it can like differentiate into like neutrophil, eosinophil. And other cell types. So what's the triggering like? Is there any, let's say, this specifically like fate determination into neutrophil are regulated by, let's say, the inflammation conditions?
Yeah. Okay. So for to answer you in a very simple way is that we don't know at this stage, but while we don't know, but it has some kind of a data or background that may help us to further explore this question is that what we found is that in our paper, the 2020 immunity paper, we did actually show that in response to inflammations, like you said, why the when or how neutrophil decide to become neutrophil. So we show that in terms of sepsis, we use a CLP model, it's a sickle ligations function model. In terms of sepsis, what we found is that there's this balance between the monocyte and the neutrophil, and this balance is very very important. So what we observe is that in a CLP early phase, all the kind of a commitment will skew towards neutrophil, and if this mouse cannot successfully kind of balance it back to the monocyte, those mice will not survive. They will die. Whereas for those mice that can kind of come back to the monocyte phase, then they will survive. And because of that, we also so-called inhibit this skew commitment by injecting anti-GCSF. Because GCSF is a key factor for neutrophil differentiation. So we injected anti-GCSF, try to skew it back to the monocyte, and this actually help to kind of the mouse to survive. And to your question, to directly answer your questions, that do we know what factors or transcription factor? We don't know, but because of now we know the ProNeu1, ProNeu2, the early phase of commitment. In fact, our RNA-seq data set can help us to determine that. That is also, there's one or two candidates that we think could be the transcription factors that that can help to can kind of determinant kind of factors that neutrophil commitment will start. But we did some experiment whereby we tried to transfuse and then Cas9, this kind of cell line and things like that. We did not go so well because we are not a molecular lab. Because like I mentioned, Xiaoyi, you may know that I was trying to apply to Tsinghua 1 or 2 years back. I was trying to go there because I really want to do some molecular work, and Tsinghua Immunology Institute is really strong at that. And these are something that we're currently doing, and hopefully in the future, we can pinpoint out 1 or 2 of these transcription factors that is a deciding transcription factors for neutrophil commitment. So that is something that we can do.
Okay, that's great. Maybe I have a last question. So about the immature versus mature neutrophils in your like laser injury model, which both get mobilized. So functionally, so is there any difference like in terms of degranulation or undergoes these netosis, like they release these DNA trap to like serving as a like inflammatory like pool, or like, so is there any functional difference between the immature and the mature neutrophils? Yeah.
So for us, we have looked at the definitely some we use some in vitro assay to address some of the functional differences between them. So we have performed the transfer migration assay. The immature one migrate less than the mature, And also we look at the ROS production. The immature one produce less ROS. And then the NATOcysts we haven't done because somehow my lab just cannot get it to work, but there are so many other labs can get it to work. But there's a recent eLife paper showing that, if I'm not wrong, they show that the immature one can release more nets. So there's a new paper showing that. And what else did we look at functionally? I think we look at the Transwell, ROS. I think that's about it. That's about it that we looked at so far.
I see. Okay, that's good. So like the marker difference between matured and immature. So what will be, if we want to say, okay, this is matured versus immature, what will be the best marker?
So this is one of the things that very commonly used in the field now. I shouldn't say commonly, like the people that really carefully do, they will use it, is CD101.
CD101.
Yeah, CD101 positive, they will be the mature neutrophil. CD101 negative will be the immature. Because the rest of the other markers that we look at, they will be regulated again by the inflammation. They will be either up or down by inflammation. But CD101 is super constant. It only expressed in the mature neutrophil. And excitingly, we recently just got the mouse for this FLOX101. So we will be able to specifically target a mature neutrophil, hopefully in the future.
Wow, that's really exciting. Okay, thank you so much. Yes, as Xiaoyi said, like I'm fully motivated.
We do have 2 more questions. I think probably we would give 2 more questions from the chat box actually. And I think one from Cheng in Philly, right? And the Cheng question is, are all the tumor-associated neutrophils have MDSC activity?
Okay, so this is a good question. There are many labs, especially Grabovich lab is used to be in Vista. I think he's no longer there. He showed quite a bit of this tumor neutrophil will have a suppressive activity. I do believe that the tumor-associated neutrophil will have, if you look at them in bulk, definitely you see the suppressive activity. However, it is based on our current data, and slowly what we think is that because they are taking it in bulk, right, and from the tumor, and then do it, there's suppressive activity, then they call it all neutrophils like that. But in fact, within this bulk population, there will be just one population is doing maybe 90% of the suppressions, and then the rest of the population is just there as a bystander neutrophil. So we do believe that there is a spectrum. There's a spectrum of suppressive activity. The MDSC activities, like you said, is a spectrum depending on how reprogrammed they are. So in our case, we can slowly dissect it out if our legend screen data allow us to really tease out the totally reprogrammed one based on certain markers, and then those that are not so reprogrammed, then we can really look at all these spectrum. Yeah.
Right. I think despite of, really intense interest in neutrophils, we do have to release like Guan at certain point. So I think I will read out one last question in the chat box. And I think if people want to interact with Lai Guan, I think probably they can do it in private. And I'm sure you have a lot to talk about. I think the one last question from Tong Biao is, are there any crosstalks between tumor-infiltrated neutrophils and the macrophages? Do they cooperate to deal with tumor?
Okay, so this is very interesting. We don't know, but based on some of the previous work that I was involved in, especially during my postdoc days with my former mentor, definitely at least the macrophages will kind of regulate the recruitment of neutrophil and providing some sort of factors for neutrophils. So I would think that definitely there will be interaction, especially macrophages, as we all know that it play a big role in tumor genesis, but we don't know. And I hope that all these good, I will take all these good comments and questions from you guys and work on it. So I'm truly appreciative of you guys helping kind of doing my revision already. Even the project is not ready yet. Thank you so much. Yeah.
Yeah, thank you again for a wonderful talk. And
so I just want to quickly have my WeChat up. Yeah.
Yeah. For I saw the WeChat is in the chat box, and everyone's welcome to connect with Guan and to think more about neutrophils, right? And now today is the day of neutrophils in spotlight. Okay, thank you. I think bye bye.
Okay, thank you so much. Bye bye. Thank you.
Yes, thank you, Guan. Thank you, Xiaoyi.
Thank you, Xiaoyi.
Yeah.
Thank you, Xiaoyi. Thank you so much.